Hello everyone, welcome to today's lecture. Today we'll be learning about sterilization. Sterilization is a fundamental and essential process in microbiological experiments, so it is crucial to understand it. In this lecture, we will cover the following topics about sterilization. First, we will define what sterilization is, and then we will discuss two common sterilization methods, autoclave and filter sterilization. We will learn about the principles and characteristics of each method and focus on how to use the autoclave and steady HEPA filter or filter sterilization. Sterilization refers to the physical or chemical actions or processes that destroy or eliminate microorganisms. There are various sterilization methods such as autoclaving, filtration, EO gas sterilization, and dry heat sterilization, plasma sterilization, and chemical liquid sterilization. Today, we will focus on autoclaving and filtration sterilization, which are frequently used in microbiological experiments. Why is sterilization necessary in microbiological experiments? Without sterilization, unwanted, contaminated microorganisms can grow and affect the results of microorganism we want to experiment with. Therefore, sterilization is essential to prevent unwanted microbial interference and ensure the accuracy of the experiment. Let's look at autoclave sterilization, a high-pressure stem sterilization method often called the autoclave. The autoclave is shown in the image below. Inside this sealed device, high pressure is applied to generate steam at temperatures above 100 degrees Celsius. It is sterilized by using pressurized steam at 121 degrees Celsius for 15 to 20 minutes. The reason for sterilizing at 121 degrees Celsius is that at this temperature, microbial spores are destroyed. Spores are highly resistant structures formed by microorganisms to survive extreme conditions, including high temperatures or chemicals. They can survive in harsh environments, and when the environment becomes less extreme, spores germinate into normal cells. Therefore, it is necessary to remove spores at 121 degrees Celsius to fully eliminate microorganisms. Now let's take a closer look at how to use an autoclave. To open and close the autoclave, you need to use the handle. Turn the handle in the open direction to open it and check if there is enough distilled water inside. Next, prepare the materials to be sterilized. To sterilize media or flask, wrap the openings with aluminum foil. Be sure not to seal the foils or lid tightly, as this could cause an explosion due to pressure. Attach sterilization indicator tape to the materials. After the sterilization process, the tape will change color, confirming that the sterilization has been completed. Once everything is prepared, place the materials inside the chamber, close the lid, and turn the handle to the closed direction to seal it. On the autoclave panel, you can check the current internal temperature and set temperature. You can adjust the temperature and time through the panel controls. The actual sterilization time is about 15 minutes, but due to the pressure increase and decrease inside the autoclave, the process can take over an hour, so plan accordingly. Once sterilization is complete, the machine will stop. After it fully cools down, open the lid and use the sterilized materials in your experiments. Be cautious of hot stem when opening the lid. Autoclaving is effective for sterilizing microorganisms using high temperature stem, but heat-sensitive materials like antibiotics, proteins, and some plastic materials may have their properties changed or degraded in such an environment. In these cases, filter sterilization should be used instead. Filter sterilization uses filters with pores smaller than the size of microorganisms to remove them. Filters look like the image on the left. Generally have pore sizes ranging from 0.2 micrometers to 0.45 micrometers. In the image on the right, you can see the bacteria and fungi, which are larger than the filter pores, can be sterilized, but viruses and very small bacteria like Mycoplasma can pass through the filter. One filter sterilization method is using HEPA filters. HEPA filters can remove fine particles and contaminants from the air. A clean bench, which uses HEPA filters, is an essential piece of equipment in microbiological experiments. In the clean bench, sterilized air is supplied, protecting the samples from external contamination. It is essential for experiments involving microbial culture and aseptic operation. In this lecture, we learned about the importance of sterilization in microbiology and explored two common methods, autoclave and filtration. These techniques help prevent contamination and ensure accurate experimental results. Proper sterilization is essential for maintaining reliable and reproducible outcomes in microbiological research. Thank you for your attention.